Good morning and good afternoon. Thank you for joining day 3 of our 2020 virtual user meeting, ligand-gated ion channels and drug discovery. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's presentations. By default, all participants are muted, and upon completion of the presentation, I will open the floor to some questions. Please do not hesitate to submit your questions throughout the course of the presentation in the chat box. It is now my pleasure to introduce Dr. András Horváth, Application Scientist at Nanion Technologies, presenting measurement of transient receptor potential cation, otherwise known as TRP, channels using the patch liner and PortaPatch. I'll now hand over to Dr. András for the start.
Thank you, Jason, for the introduction, and good morning to everybody. And welcome to also on the behalf of the Patch Liner and PortaPatch team to our virtual user meeting. And as Jason mentioned, I'm going to talk about recording transient receptor potential channels using the Patch Liner and the PortaPatch. So my name is András Horváth, and I'm working both for the Patch Liner and the PortaPatch team. So first of all, I would like to start with the machines which I used for this this studies. So first of all, the PortaPatch. As many of you might know, this is our smallest single channel patch clamp device. It is easy to use and learn. You can perform stable gigascale recordings with it. There's small footprint. There's low compound consumption. You can use it either in wholesale, cell attached. You can do wholesale, cell attached, and wide area recordings. You can use it in voltage and current clamp mode. And it has practical add-ons such as the external temperature controlled perfusion, which I'm going to introduce more detailed in the next slides. We have a very unique internal perfusion exchanger on it, and also microscope slides. So a little bit introducing the external perfusion system. So on the right-hand side, here you can see the PortaPatch installed with the external perfusion system. It's temperature controlled, which is controlled by an external temperature control device, which allows you to increase, start the cool down it to 10 degrees and heat up to 50 degrees. The whole measurement, it has a very fast exchange time. It has a temperature controlled Faraday top and a temperature controlled cannula to perfuse the heated solution. You can see this temperature controlled cannula here. It has continuous perfusion due to continuous waste removal. So as you can see, we have a perfusion panel here, and there is a pump which is connected to a bottle. So it's fully also automated. You can perfuse up to 8 different solutions. You can have real-time information about the bath temperature because we have an integrated temperature sensor here. And you can trigger perfusion manually or software controlled. And here is a nice application of the perfusion, of the fast perfusion system. Here you can see a ramp recording on the TRIP-M8 channel activated by increasing concentration of menthol. So this is, and as you can see here, 5 concentrations could be applied at one time, each and after, of course. So the other system I used is the Patch Liner. It's our fully automated 4-channel or 8-channel device. It has important features such as the external and internal solution exchange. It has a temperature control, so you can apply it for heat activated channels, which is going to be also one of the application I will introduce later in more detail. You can do voltage clamp and current clamp recordings, and it has a special feature that you can also use the dynamic clamp interface for it, which is also fully automated. It has single and multi-hole chips, which are produced here in Munich. It is doing series resistance compensation. You can do recording at individual V-halves. So you can measure voltage and ligand-gated ion channels, wholesale and perforated patch clamp recording could be performed. You can measure cell lines, primary cells, and stem cells. You can do action potential pharmacology, a very unique feature, I think, for this machine. It has minimized cell consumption, validated for SIPA, and it has easy and customizable analysis tools. So one of the special features of the patch liner, which is suitable for transient receptor channels, is the application of heated solution. So as you can see, the solution is heated in the pipette until it reaches the desired temperature. And as you can see, and then as a heated solution flows over the cell, the 3-3 channels are activated. You can see here also on this original recording and also on the time course recording. Also, when the solution cools back to room temperature, the 3-3 channels return to baseline. This is what you can see here. So a little bit about the experimental conditions. What I did, it's pretty a full slide, but let's just go quickly through it. So we were using CHO cells expressing the 3-3-1, 3-3-3, 3-3-4, and 3'8 channels. In case of 3-3-1, I used 10 micromole capsaicin and heated external solution to activate the channel, and 10 micromole capsazepine or ruthenium red was used to block the channel. In case of 3-3-3, 2 APB was used and also heated external standard solution. In this case, ruthenium red was used to block the channels. In case of 3-3-4, the GSK agonist, 3-3 channel agonist was used and also heated pipette. And to block the currents, the ruthenium red and the GSK antagonist was used. In case of 3'8, we activated the channel with 10 degrees of external standard cooled solution, and the block was performed by 50 micromolar capsazepine. Here you can see that we applied a classical REM protocol from -100 to +100 millivolts. Yes, and also the average incubation time was around 60 seconds per compound, which we found enough because of the rapid solution exchange. So I would like to show the results. First, the transient receptor potential cation channel number 1, so the 3'1. This was performed on the patch liner. And as you can see on this slide, on the left-hand side, you see the original recording, and the blue trace shows when the 45-degree external solution was applied. And as you can see, the time course, that there was a huge and very rapid activation of the channel. And interestingly, the ruthenium red, pink, so the heated solution together with ruthenium red, we found it interesting that still we could activate the channel with temperature. And we also applied pharmacology on the channels. So as you can see on the original slides, original traces, the capsaicin is rapidly and activates a robust current, which was sensitive to ruthenium red. Also, in case of capsazepine, first we activated a large current, and then we were able to block it with both blockers. And on this bar graphs, you can see that in 5 experiments, we were able to activate a robust current at the outward part, which was sensitive to capsazepine. So as a next target, we tried the 3'3 channels, 3'3 recordings. So as you can see also on the upper left, that the heated solution could repeatedly activate the current. Also, you can see here on the time course recordings, very nice activation. And also interestingly, the ruthenium red, even at very high concentration, was not able to prevent the activation of the current. However, as you can see, which we saw also in the literature, that the basal current during the experiment was increasing, and interestingly, the ruthenium seems to have an effect on it. Also, in case of pharmacological activation, the 2-APB activated a large outward current, which was sensitive to 200 micromolar ruthenium red. And also, we repeated those experiments in 6 times, and as you can see, compared to the TRPV1, there was already a larger basal current, but still we were able to activate a very large outward current with the 2-APB. In case of TRPV4, we applied this, we tried both on the patch liner and the PortaPatch to activate the current. So as you can see, the heat activation also worked on the TRPV4 channels. Also, the pharmacological activation seems to be working. However, it seemed to be slower compared to the previous channels. So we see compared to the others, a slowly activating current, but it seemed to be sensitive also to the antagonist. And as you can see, we were able to activate a large current with the GSK agonist in 7 recordings. In case of PortaPatch, we also successfully applied 45 degrees heat activation with 45 degrees. Compared to the patch liner, it was more like activating slower, but interestingly, it was sensitive to ruthenium red. And in case of the pharmacological activation, we were also able to activate the current nicely, and it was sensitive to ruthenium red. So, and for the PortaPatch, we applied cold TRPM8 channels. We tried TRPM8 channels, which are cold activated. This is one of the unique features of the PortaPatch, that cooling down the housing and the whole experiment to 10 degrees, we were able to perform it. And as you can see also on the original recordings and on the time course recordings, that at 10 degrees, we were able to activate a very nice current. And when I heated up the experiment, then the current got inactivated and it could be repeated again. And when I applied 50 micromolar capsazepine, the current was blocked and we were able to wash it out. So we can say that cold activation could be repeatedly applied here, and the capsazepine was effectively inhibiting it. So that was a very nice and exciting experiment. And in conclusion, both the patch liner and the PortaPatch are powerful tools to study TRP channel physiology, also pharmacology, we can say that. In case of the patch liner, using heated pipette is a unique tool to study heat activation. In case of the PortaPatch, its fast and temperature controlled perfusion system can be used to study heat and also cold activation. And both devices are suitable for ligand activation and could be used to find compounds which block the temperature and ligand response separately. So with this, I would like to thank you for your attention, and I'm happy to answer the questions. Thank you.
Thank you so much, András, for going through the data. Again, I'll open up the floor to some questions now. Initially, the first question, András, regarding TRPM8, are you planning to do ligand activation?
Yes, I didn't show it yet, but this is already tried, but of course we're going to repeat it, and we will do it more detailed how to apply it. So yes, we are going to do it.
Then another question comes in regarding the temperature range. What the temperature range is for the patch liner? Does it cool? And what is the maximum temperature you can heat to?
So basically, regarding the temperature, you can heat up, I think, even to 60 degrees, which I was playing a lot with the temperature during the 3P studies, but there is one point where the cell just dies if you apply too high temperature. So I would say you can go up way more higher, but in this case, I would not necessarily recommend it because it can also damage the cell.
Mm-hmm. And then did you try to activate TRPV4 with a higher temperature than 45 Celsius?
I tried that, but as I mentioned before, it was quite difficult because the cells did not tolerate that well the higher temperature. But of course, I can try it again. But in my case, and this runs too high temperature was a little bit too much also for the cells.
Okay. And then again, for TRPV4 ligand-gated activation, the activation was slower. Could you elaborate more on this?
Yes, that's a good question. I mean, I was a little bit surprised that it was slower, but I don't really know why it happened. Maybe it's a feature of the channel, but I mean, also on the PortaPatch, it seems to be slower. So I don't think it's any machine-related thing. So I would say it's somehow maybe a feature of the channel that it's activating slower. So, and they were different kind of cells, so not the same source. And in both cases, the activation seems to be a bit slower than the 3P1 and the 3P3, for example.
Okay. Then another question, have you tried liposome patches in the PortaPatch?
Yes, so liposomes could be done. I mean, it's of course not the easiest application, so you need a good preparation for it. But it can be done. We have also application notes about it.
Okay, perfect. So then what I'll do is I'll make sure that the recording of this presentation is available on demand. That usually will be by the next morning. Any other additional questions, we welcome you directly. I'll make sure to include András's contact details in the description. And naturally, if you have any questions regarding this presentation or the technology used, feel free to get in contact with us at the next convenience. Our next talk will start at the top of the hour, and we thank you for joining us for day 3.
Thank you.